like, damn it, lady. Okay, I think I'm live. All right, let's do this. This is chapter BIO116, Anatomy and Physiology 2, chapter 14, which is about blood. You can see here in this blood vessel, there's a lot of red blood cells. So what's important? Of course, know the functions. Here's some nice ones. Stability of interstitial fluid, distribution of heat, homeostasis, big thing. Know your different blood cells and what is their function. Red blood cells, also known as erythrocytes, white blood cells, leukocytes, platelets are thrombocytes. Remember, platelets are simply cell fragments, and know the function of each. Red blood cells carry along with hemoglobin, and white blood cells, part of the immune system. Platelets is part of hemostasis, or the blood clotting function. Next slide, this is beautiful because it situates the formed elements, which is the dry part of blood, versus the plasma. You see there's 45%. That is your hematocrit, or the ratio between formed elements and plasma, which is the hydration status of your patient, hematocrit. Now, your CBC, your complete blood cell, counts the complete number of platelets, red blood cells, and white blood cells. But your white blood cell differential, or your diff, has the exact percentages of your neutrophils, eosinophils, basophils, monocytes, and lymphocytes. The first three are your granulocytes, and the last two are your agranulocytes. Plasma, the fluid part of your blood, of course, has electrolytes. It's made out of water, tons of it, and of course, it has proteins. The albumins are for water balance. Globulins or immunoglobulins, part of your immune proteins, and your fibrinogen is also aids in your platelets in clotting. Nutrients, vitamins, and hormones are also in your plasma. The three main gases in your body, nitrogen, oxygen, and carbon dioxide. Remember, the chemoreceptor in your central nervous system in your cerebrospinal fluid is the main monitoring chemoreceptor is carbon dioxide. Plasma here, RBC, hematocrit here. Morphology of your red blood cells here. This is biconcave disc. Here's some granulocytes here. I'm going to talk about which one is this one, which one is that one, which one is this one. And of course, the platelets are cell fragments from a megakaryocyte, and these are sticky fragments that are good for clotting. You'll have the spun or centrifuged blood, right? And if it's maroon colored like this, you know that this is venous blood. And this is venipuncture that's going on right here. Stem cells are anything that's hemocytoblast or poiesis. Remember, hematopoiesis is in, or the formation of the formed elements is in the red marrow of your blood. You have lymphoid stem cells in the red marrow will give rise to lymphocytes. Myeloid will give rise to your white blood cells, red blood cells, and platelets. So myeloid, think everything, lymphoid system, just your white blood cells. Well, particularly your lymphocytes, which is your T and B cells. Red blood cells we already talked about as they are anucleated, means it has no nucleus, but in the middle they have hemoglobin. Oxyhemoglobin versus deoxyhemoglobin, also known as carboxyhemoglobin or hemoglobin that carries carbon dioxide. Biconcave disc, as you can see here, this is a concave section. There's another concave section. There's two of them, hence the term biconcave disc. There's no nucleus, therefore there's no mitochondria, right? Therefore they don't divide, they just die off every 120 days. And you make new ones inside your red marrow in response to erythropoietin, which we'll talk about momentarily. Oxygen carrying capacity is the signal for your liver and your kidney to make erythropoietin. If you have low blood oxygen, your liver and your kidney will sense that, just like the way chemoreceptors sense low blood oxygen. Your chemoreceptors in your liver and kidney, if they sense low blood oxygen, it will activate erythropoietin, which will travel through your bloodstream and then get into your red bone marrow so you can make more blood cells. Once the carrying capacity starts increasing, there will be a negative feedback to this system and will tell the liver and kidney to stop releasing erythropoietin. We mentioned sickle cell anemia here in class. Remember, abnormal morphology leads to abnormal things. Phagocytosis is by macrophages, and your red blood cells naturally die off in 120 days or so. And in the middle of your red blood cell is your hemoglobin, which you can see there are 4 heme groups, which is an iron-containing group, and they're kind of like chairs where the oxygen and carbon dioxide get to sit on. And the attractive feature is this iron in the middle of, also known as your heme group. You will also notice here the breakdown of red blood cells in both your liver and your spleen, but mostly in your liver. Hemoglobin will break down into heme and globin. The globin part, or the protein part, will break down into amino acids, and then your heme part will break down into iron and biliverdin. The biliverdin will then get converted to bilirubin in your liver. Bilirubin will then go into your blood and then end up in your kidneys, while any excess biliverdin will end up as bile in your small intestines here. Granulocytes versus agranulocytes. Know that neutrophils, eosinophils, basophils, granulocytes. Agranulocytes, lymphocytes, and monocytes. Know these terms: diapedesis, cellular adhesion molecules, phagocytosis, inflammatory response, and positive chemotaxis. This is a nice picture here of diapedesis. Dia, complete or thorough. Peds, walking. What are we doing? We are completely walking out of the capillary or the blood vessel, and this is a white blood cell, and it's walking out and it's being attracted via chemotaxis to a site of infection, where the white blood cell will then adhere or stick to the injury or the area of concern, and then will perform phagocytosis by eating and digesting a potential pathogen. Know the difference between your differential, neutrophils, eosinophils, and basophils. They are all part of your granules, granulocytes, and your neutrophils, also known as PMNs, first one to fight, very strong phagocytotic capabilities, and they're elevated in bacterial infections. Eosinophils, they have a bilobular nucleus versus your multilobular nucleus here, as pictured here. Multilobular, PMN, neutrophils. Bilobular here, this is one lobule, this is another lobule, and that's what? Eosinophil. Also good for 2 things. Parasitic infections and allergies. So that's eosinophils, bilobe nucleus. They're good for 2 things: allergic reactions and parasitic infestation. Monocytes are the largest of the white blood cells, and they can turn into something even larger called your macrophages. They're considered granulocytes. They have granules here, but they're not as pronounced as the granulocytes here. So this is a monocyte. And this one here is a basophil. Now, what's so important about basophils? Along with mast cells, they release histamine and heparin, and histamine enhances inflammation, and heparin is a blood thinner, or it'll help the blood stop clotting a little bit so that the blood can get to the area of infection. Lymphocytes, B cells versus T cells. The T cells are from the thymus, B cells are from your bones. The T cells are T helper and T cytotoxic or T killer cells, and the B cells are, once activated, they form antibodies. Therefore, the B cells are called memory cells because they make antibodies. Know this term, leukocytosis, leukopenia. You can also apply it to your other formed elements, erythrocytosis versus erythropenia, thrombocytosis versus thrombopenia. Thrombopoietin is what is produced by hemocytoblasts to make clotting cells, or thrombocytes. Thrombocytes are simply fragments of these cells called megakaryocytes. These megakaryocytes break apart into small cell fragments that are very sticky. And they provide clotting or stopping the bleeding, also known as hemostasis. Serotonin is also released because it has the effect of smooth muscle to contract. The smooth muscles in your blood vessels, especially your arteries, will then close it off, and then there'll be less bleeding going on, less blood loss. And that's serotonin, abbreviated 5-HT. Blood plasma, we already talked about. We already talked about plasma proteins. And there are proteins that are not an energy source. The most important blood gases in your plasma is oxygen, carbon dioxide. Nitrogens are not so important because you have a vast abundance of them in your atmosphere. There's some other nutrients. Know and remember electrolytes. Some of them turn things on, some of them turn things off, but there must be a balance. Hemostasis, another fancy word for blood clotting. The blood vessel vascular spasm, that also has to do with serotonin, and your tunica media of your arteries, which is your middle layer that helps constrict blood vessels, especially the artery. Blood coagulation has both extrinsic and intrinsic clotting factors, and these factors, along with vitamin K, these chemicals here, they regulate blood clotting, so we don't have too much, don't have too little. Fibrinogen is also part of the mix because fibrinogen is the precursor to the protein fibrin, which makes a whole bunch of insoluble threads along with all these platelets, makes a very sticky net so that it can prevent any significant blood loss. Know what serum is? We already talked about that. Plasma minus the fibrinogen, right? Has most of the clotting factors, platelet-derived growth factor, right, along with serotonin, stimulates smooth muscles, but along with platelet-derived growth factor is more about repairing damaged blood vessels. And plasmin, of course, digests the fibrin threads. So it's like the opposite of fibrinogen. Fibrinogen makes the fibers so that you have a thicker clot, but the plasmin then dissolves the clot when it's no longer needed. Know these terms as well. This is slide 36, thrombus versus embolus, which one moves, which doesn't. Of course, the embolism is much more dangerous because it moves around. And atherosclerosis, which is the cumulative fat in your arterial lining. And here's an example of which, because that artery could get hardened, and then we'll have a hard time dealing with the blood pressure or the increasing blood pressure that ensues. Know your blood types. Remember ABO versus RH. Know this chart. Definitely it's going to come out. And if you need to know the meaning of the antigen versus antibody, remember the antigen is what is on the jacket. So if you're in gang A, what are you going to have on your jacket or the coating of your red blood cell? You're going to have an antigen. And of course, A's don't like B's, so you're going to have antibodies, that Y-shaped protein that will eat up what? Anything that's B. Universal donor, O. Universal recipient, AB. Here's an example of glutination. Here's an example of antibodies. Here's your peripheral smear with a whole bunch of white blood cells. Those are those purple ones. And you can see the PMN here. You can see a monocyte here. This, I'm not quite sure. It might be a lymphocyte. And these little specks of dirt, these really light ones like this one, that's a platelet. This is also like that. And of course, the majority, red blood cells. And last but not least, RH blood group deals with Erythroblastosis fetalis, also known as HDN, or hemolytic disease of the newborn. It's when the Rh positive fetus is carried by an Rh negative mom, and it's not the fetus is the first one to get sick, it's the second one. The second Rh positive baby will have something called kernicterus, which is an extreme form of jaundice, where even not only the skin gets painted with bilirubin, it is also the brain gets painted with bile pigments. And that concludes chapter 14 video. Hope to see you in class. Bye.